Welcome to the Chemistry, Biology and Math Revision Hub. Today we're doing the A-Level Biology topics. This is Gene Technology, beginning with topic 8C1, producing recombinant DNA. To produce recombinant DNA, we need a gene of interest, for example that, and we need a specific vector to help us to deliver the gene. In this case, this plasmid is going to be the vector. So we can use the stem restriction enzyme to cut out the gene of interest from the DNA, as well as to cut the plasmid so that we leave complementary sticky ends. We can see this sticky end and that sticky end are complementary. So when we combine the two together using DNA ligase, it will be easier for the gene to be inserted. So now we have created our recombinant DNA through the combination of the gene of interest as well as the cut plasmid part. This is going to be introduced into the microorganism, in this case a bacterium, and now the organism produced could be a transgenic organism since it has DNA coming from something else. Sometimes recombinant DNA can be made using messenger RNA, where the messenger RNA is converted into complementary DNA, and then that DNA could be inserted into a specific organism. And like I said, you have to use a stem restriction enzyme because a restriction enzyme cuts at a specific recognition site. So the plasmid and the gene of interest are going to be cut at the same section or at sections that have the same base sequences, so that sticky ends are left that are complementary, so that when DNA ligase is used, we can be able to create or easily attach the two together for the creation of the recombinant DNA. The next part is to identify if an organism has been transformed, meaning if it has successfully received the intended DNA. To do this, we could use a method called replica plating. Replica plating involves pressing a sterile velvet cloth onto the master plate to pick up some cells from each of the colonies, then pressing it onto a secondary plate with selective media. Then you will incubate. After incubation, growth of the replica plate is compared with that on the master plate to identify colonies with specific traits. This could have antibiotic resistance or ability to produce some metabolites. So in this case, this is the master plate. And then we see the sterile velvet, which is going to be pressed onto the master plate. And then that velvet is going to be pressed onto another plate, onto which imprints are going to be formed. These are the imprints of the colonies, and that is going to be cultured. Then in the end, we're going to compare the cultured plate or the replica plate as well as the master plate to see which colonies are existing. For example, this colony, that and that, are present on the master plate, but they are missing on the replica plate. These are the colonies of the organism, like in this case, that were transformed. So we can be able to pick from that specific colony or the two colonies and culture, because those are going to be the microorganisms that were successfully transformed. Vectors are used to deliver the genetic material into the organism that is intended to be transformed, and this can be viral or non-viral. The viral vectors involve using viruses that have been weakened or that are not going to cause any problems, while the non-viral vectors include the gene guns, which shoot nanoparticles coated with DNA into cells at high speed. They include the liposomes. They surround the DNA, forming a bilayer, and they will fuse with the membrane in order to deliver the genetic material into the cell. There is also microinjection. Here the genes are going to be introduced by injection into the specific cell using a micropipette. And from current studies, it's been established that viral vectors are better at taking the DNA material into the nucleus in comparison to the non-viral vectors. During genetic modification, knockout organisms can be created. Here, genetic modification is used to silence genes, and a new gene is going to be inserted in a gene to be investigated. This is going to make the original gene impossible to transcribe, and so it cannot be used to make a protein. The process of knocking out genes can be used to find out the functions of certain genes, because their absence will lead to deficiency of certain molecules. So we can be able to find out, since these molecules are missing, it's because that gene was responsible for the production of those specific molecules. Next, topic 8C2, drugs from genetically modified organisms. Genetic modification uses microorganisms because the microorganisms are cheaper and they're easy to culture. They reproduce rapidly. A transfer gene is going to be copied rapidly, and bacteria can be cultured on large scale, yielding large amounts of biological molecules within a shorter period of time. The microorganisms can also be used to make biological molecules like insulin, growth hormones, vaccine, and so on. In talking about vaccines, there is potential of use in banana vaccines to overcome logistics related to vaccine storage, like refrigeration. During insulin production, the human insulin gene is going to be isolated. Restriction enzymes are used to cut out the gene from the DNA, and the same restriction enzyme is going to be used to cut out genes from a plasmid, leaving complementary sticky ends, like we already said, and DNA ligase is going to be used to insert the gene into the plasmid. We can see that. So here we have cut out the gene of interest, which is the human insulin gene, and then the plasmid is also cut out. Combining the two using DNA ligase, we create the recombinant DNA, and this recombinant DNA is going to be introduced into a bacterial cell or bacterium. Using this transformed bacterium, culture in fermentation tanks is going to be carried out in order to produce many bacteria of similar kinds. Then the insulin is going to be extracted and purified in order to produce a human insulin. Going on to making transgenic plants. Plants can also be transformed to obtain specific traits that are beneficial. A good example is using the bacterium A. tumefaciens, which has ability to infect plants. It has a specific plasmid, which we can see here, and this plasmid can be extracted out. Certain genes can be cut out, and the genes of interest can be introduced, like we see here, the new gene. And after, we can return to the bacterium, and then the bacterium containing the plasmid is going to infect the plant, introducing the genes, which are going to be used to make biological molecules within the plant, or which are going to be used to give the plant a specific advantage. The cells that are going to be produced in here will have that specific DNA or the gene that has been introduced. If we take off some cells from the crown ball, under specific conditions, we culture those cells, allow them to grow into specific plants. The plants are going to contain the specific trait or the gene that was introduced in them. And now we have transgenic plants, meaning plants that have genes from other organisms. Also, we can create genetically modified animals. In making transgenic animals, human genes coding for specific protein are going to be introduced into the egg of an animal. This other gene will have its accompanying promoter sequence to ensure that it's going to be expressed in the mammary glands of that specific animal. If the egg that received the gene is fertilized, then an embryo is going to be formed, and this embryo is going to be implanted into a surrogate mother. After delivery, the animal is going to have the ability to produce a human protein in its milk. Moving on to topic 8C3, microarray and bioinformatics. Microarray is used to determine which genes are expressed in a cell of an organism. To do this, messenger RNA is used to produce complementary DNA or cDNA. And cDNA is given a fluorescent label, which is usually green. And the known sample, meaning the sample we're comparing with, is going to be given red. The labeled samples are going to be mixed together and then applied to a microarray slide. And the added labeled cDNA hybridizes with the probes. The microarray is scanned to measure fluorescent light produced by the different spots. If both samples are expressing the genes, the color is yellow. And if the experimental sample expresses more than the control, it will appear red. If the experimenting sample expresses less than the control, then it's going to appear green. In disease only, we can see green. In normal cells only, and if both, then we can see it's going to be yellow. Most of the time, bioinformatics is going to be used in the interpretation of the results. It uses algorithms, mathematics, statistics, and so on to interpret. Moving on to topic 8C4, the benefits of genetically modified organisms. Here we're going to look at the benefits of genetic modification of crops. Genetic modification can give pesticide resistance. Here plants can be engineered to make self-made pesticides within their leaves. We could produce flood-resistant rice. This will save a lot of rice from destruction in areas where flooding is very frequent. Genetic modification can enable us to change the nutrient value of plants. Here the content of the macro as well as the micronutrients can be altered to provide nutrients that are suitable for consumption. We can also produce herbicide-resistant plants. This is usually done in soybeans to produce plants that are resistant to commonly used herbicides, so that basically when the herbicides are sprayed, the soybeans are not going to be destroyed. And then we can do that for fatty acid balance. You remember the monounsaturated fatty acids are better or healthier than the polyunsaturated fatty acids. So if we can create a balance between the two in a specific food sample, then the consumers are going to get a healthier meal in order to overcome certain diseases. And there is potential of using genetic modification in therapies to overcome health issues like cystic fibrosis. If there is a possibility of altering the genetics of an embryo, then we can be able to produce an individual who does not have the genetic condition. Next, we look at the risks and benefits of genetic engineering. We can see antibiotic resistance. This is a risk because some organisms could obtain antibiotic resistance in nature, and if these are pathogens, it means available antibiotics will no longer be effective in curing diseases caused by those pathogens. And because in genetic modification, sometimes plants produced do not have seeds or they're infertile, then the farmers have to depend on the scientists to provide the seeds during every planting season, and this could be unfair to people from poor countries or farmers from poor countries. It could also encourage monoculture. This is because if one plant is successful and many consumers like it, then other plants will not be cultured, and that is going to lead to the extinction of certain species of plants because one specific plant or crop is going to be grown. It could also lead to creation of superweeds. This could be due to maybe horizontal gene transfer. A certain genetic advantage is going to be gained by weeds, and the weeds will no longer be destroyed by the herbicides. Then the weeds are going to grow and outcompete the plants in some areas, and this will not be good for farmers. Then also there is this risk of engineering humans, like designer babies. Of course, this is not a threat right now, but people who look into the future, if genetic engineering is allowed to be carried out on humans, then there is a chance of engineering babies to produce designer babies, which could be a problem. There is also a possibility of horizontal gene transfer. Here, this is unintended, but genes could be transferred between different organisms in nature, and this will have unintended risks, which we do not know or which we cannot predict. Some people do object using research on animals because in some cases it's going to be unethical. And that because of the financial imbalance in different countries, the gene technology could be properly developed in a few countries, and in other countries they're going to have problems because it's not going to be well established. The benefits are we're going to have better food production, meaning a lot of food can be produced within a shorter period of time in a small area in comparison to without genetic modification. There is development of medicines like those for diabetes. This is already available and we have seen the benefits from it. There is potential use in gene therapy like we see in cystic fibrosis, potentially altering the DNA so that we can overcome certain genetic diseases. And there is a chance of production of large quantities of biological molecules like enzymes at a cheaper cost, and the time frame is going to be short. And that will be beneficial to people who will be lacking those biological molecules or those who use them in specific processes of their life. So this brings us to the end of this video, which is about gene technology. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.